All right, so antigens, as we've said, are the things, the foreign types of molecules that make you ill. So pathogens are basically antigens. These are the things that elicit some sort of an immune response. So what happens with these antigens is either some antibodies or the lymphocytes themselves, the cells that are producing them, are going to bind to what's called an epitope on the antigen. So that's the antigen binding site. That's the thing that the antibodies actually bind to. Okay, so with acquired immunity, what we were discussing here is this is what you kind of pick up along the way. And there's two main types that we've talked about. We have the humoral response, and that's having to do with the humors or the fluids of the body. And this is the circulating antibodies that are going to produce or that are going to be produced by the plasma cells. And then you have the cell mediated response, which is where you have some cytokines kind of interacting with other components of the immune system when the helper T cell binds to the actual antigen presenting cell. And we've kind of gone through these types of things, so I'm going to kind of rehash some of the stuff that we've talked about here. So with the humoral response now, one of the important things to keep in mind is that it doesn't matter if we're talking about a T cell or a B cell. Both of these types of cells are derived from the bone. So in the bone marrow, this is where they're going to grow. And then the B cells themselves, these are going to mature in the bone marrow. And the T cells are going to mature in the thymus. And they're both circulating around the body. And when the B cells become activated, and we'll talk more about the activation process here in a few minutes, they start to secrete these defensive proteins that we refer to as the antibodies. So they become these plasma cells that are secreting the antibodies, and these antibodies accumulate within the blood and the lymph, and they bind to the certain types of antigens that are, kind of floating around the body and marking them for elimination from the body. And we also get some clonal selection that occurs, and what happens, like a clone is just an exact replica. So you get clonal selection. So in other words, these B cells are going to be stimulated to make memory cells of themselves, and they're going to also be stimulated to make these plasma cells, which are going to secrete the antibodies. So if we're looking at the right here, the right part of this figure, you can see the clone of plasma cells there. Those are the things that are going to make and secrete these antibodies that will bind to these antigens that are circulating around in the body that have been kind of detected in one way or another, and then we're going to mark them for the elimination. Okay, so with clonal selection now, this is where you have the antibodies that are being secreted, and the whole idea of the ability to produce these memory copies, so that if you're infected later on with this thing, to get rid of it very rapidly, and the idea that these plasma cells are going to secrete these antibodies. This is the whole kind of fundamental, I guess, idea behind clonal selection. So these antigens, what they do is they bind to the specific part, a specific receptor on a B cell, and a small fraction of the body's B cells are going to be activated. The lymphocytes are going to spring into action, but the rest of them are going to just kind of be, at the ready for being able to respond to something else that may make you sick at a later date. And what this does is this stimulates this antigen binding to one of the memory cells is going to stimulate the production of thousands and thousands of clones. They're going to work very rapidly. Millions of clones going to work very rapidly to get rid of that antigen as fast as possible. So you can see you've got some antigen molecules here, those little red diamonds, and they're going to bind to the surface proteins on any of these particular types of B cells. And there's 3 depicted here, and only one of them is going to be activated. It's going to make more memory cells of itself, and it's going to make clones of these plasma cells, which are going to very rapidly produce these antibody molecules, which we said earlier is going to bind to and mark these antigens for removal. Okay, so let's watch this.
In 1960, Sir Frank Macfarlane Burnet was awarded the Nobel Prize for his contribution to the field of immunology. Burnet's clonal selection theory revolutionized our understanding of the immune system. To this day, Burnet's research remains the foundation of the way we think about disease and how the body responds to it. Clonal selection is the central mechanism by which your body fights infection. The following visualization presents clonal selection during an infection by strep throat bacteria. This patient has a strep bacterial infection causing a red, sore, pussy throat. The tiny round strep bacteria thrive on throat tissue. The bacteria are coated with hairs called M protein. M protein is what your body recognizes to be foreign. In response, your body will launch an immune attack against it. Your lymphatic network is an important part of the immune system, detecting any infections that arise in your body. The M protein hairs drain into the lymphatic system around this patient's neck. The lymph node is crammed full of B cells. B cells are guardians of your immune system, scanning the body for foreign material. Arriving at the lymph node are M protein hairs from the bacteria. Your body contains billions of B cells. Each B cell is unique and is sensitive to only one kind of foreign infection. By chance, one B cell is sensitive to the M protein hairs from the bacteria. This is the molecular surface of that B cell, magnified 10 million times. Its surface is covered with receptors that recognize M protein. The receptors grab onto the M protein hairs. This triggers an activation signal to travel into the cell. The activated B cell migrates and begins to replicate. In a few days, thousands of copies of the selected cell are made. This is the central idea in Burnett's clonal selection theory, where one cell is selected out of billions of B cells and identical clones have been created from it. Therefore, the cell is clonally selected. This creates an army of B cells that mass produce M protein antibodies and release them into the surrounding fluid. The antibodies circulate in the blood and travel into tissues. They bind to the M protein hairs on the strep bacteria. This flags them for destruction by the immune system. Other immune cells ingest the antibody-covered bacteria. They destroy the bacteria and eliminate the infection.
All right, so we'll watch this a little bit in class, and we'll go over it, and we'll be able to answer any questions that you might have, okay? So now, as far as the cell-mediated response occurs, this is where you get these cytotoxic lymphocytes that are directly going to destroy the infected body types of tissues, cancer cells, foreign types of tissues, and there's two main types of T cells that are going to be involved. You've got the helper T cells and you've got the cytotoxic T cells. And the other thing that the cell-mediated response is going to do is it's going to work with the phagocytes and these antigen-presenting cells, and it's going to help kind of relay the message to the B cells to kind of get them involved as well. Okay, so your T lymphocytes, these originate in the bone marrow, as we said, of course, and they mature in the thymus, and there's two polypeptide chains that are on them. You've got a variable region, a constant region, and these things are just really what's involved in binding to the cell and giving kind of the specificity, hence the term variable region. And they're linked to the plasma membrane and kind of presented out on the surface. And what these T cells recognize are fragments of these antigens that are being presented by the MHC types of molecules, either from your body cells or from the actual phagocytes. Okay, so the helper T cells, these are central to the network of these kind of cellular interactions that are going on, and they respond to the different peptide antigens, the different proteins on the surfaces, as we saw in that video. Those proteins get presented on the surface. Those are the things that are going to be presented to the helper T cells by the phagocyte, the B cell, the dendritic cell, those types of things. And what they're able to do is then release some cytokines and activate B cells and or cytotoxic T cells. Okay, so these helper T cells now, when they encounter a class II MHC molecule, and that's the kind that you find in these antigen presenting cells in terms of, or in other words, in terms of the dendritic cells, the B cells or the macrophages, they stimulate the production of clones of helper T cells and memory helper T cells, which is going to promote the removal of different, infected types of cells, different types of pathogens that might be found within the body. So for instance, if you look at the picture here, what you can see is that we have a dendritic cell here, and what it's done is it's encapsulated a bacterium. It's, chopped it up with the help of the enzymes and such inside of the lysosomes, and now it's presenting them. These pieces are binding to class II MHC molecules, and they're being presented on the surface. And now you can see that the helper T cell is going to recognize and bind to that. It's going to release some cytokines, and that's going to be the language that it's going to use to launch either a cytotoxic T-cell, if that's the pathway it's going from, like an infected type of a cell, say with a virus or something, it'll attack and get rid of those types of body cells, and we'll talk more about that in another lecture. Or, as the thing that we have been talking quite a bit about, it'll stimulate these B cells, the part of the humoral type of immunity where it's going to stimulate the production of memory B cells, as well as these plasma cells that are going to be secreting the antibodies. Okay, so what the cytotoxic T cells do now is, we'll talk about this in more detail in a different lecture, but what they're basically doing is if you have a body cell that's been, that's cancering, or it's infected by a virus or something like that, something is going wrong with the proteins that cell is producing, and it's presenting them on the surface. And as a result, all of your body cells, all of the cells inside of your body that are self cells, these produce class I MHC molecules. Your B cells, dendritic cells, and macrophages, those produce class II MHC molecules. But your body cells produce the class I MHC molecules. And what that does is those molecules bind with the foreign types of proteins that might have gotten into your cell through a virus, or that are arising as a result of a mutation that's producing them. And what they do is they bind to the class I molecule, or MHC molecules, and they're presented on the surface. And the cytotoxic T cell now recognizes that cell as being more or less in distress. There's something wrong with it. And so what it does is it secretes different types of enzymes, granzymes, things like that, perforin proteins. And what it does is it pokes holes in the cell, disrupts the water balance, and essentially is inducing apoptosis, which is the programmed cell death. So what you can see here is that you've got a cell on the left, and there's something wrong with it. The helper T cell secretes in step 2 here the perforin and the different enzymes that are going to break down the cell membrane. And then in step 3, you can see where that cell is actually kind of breaking down and being gotten rid of by other components of the immune system, being carried away and excreted from the body. And then if you look at the right picture there, the electron micrograph, that's an actual cytotoxic T cell that is attacked and gotten rid of a cancer cell. So the cytotoxic T cells, these are essentially the effectors. These are the things that are doing the dirty work of the immune system. Okay. Now we talked about the primary immune response, and we did a worksheet that kind of showed this briefly. And what you have is you're exposed to the antigen, you become ill, and your lymphocytes begin to really rapidly kind of ramp up their division, but that takes a longer period of time. And this is where you may become sick, but eventually what happens is the antibodies clear the antigen from the system. Okay, with your secondary immune response now, this is if you get exposed a second time. And this is very specific and very rapid, and this is a result of your immunological memory. And what it does is it very rapidly makes the antibodies and can very quickly get rid of the pathogen that's in your body, and oftentimes before you actually become sick. So what this is dependent upon, the secondary response, is the idea of the memory, memory of the immune system. Okay. So that pretty much wraps up everything. And if you have questions, as always, make sure that you write them down, and we'll make sure we go over them in class. All right. We'll see you soon.